secretion. Pulsatile because if you look at every hour, may mga pulses, diba? Within an hour for your ACTH and hence for your cortisol. These are short-term fluctuations, but when we smoothen and average it out, you will get a pattern of secretion of your ACTH and cortisol which are superimposed upon each other. Because once you have the ACTH secretion, this causes the synthesis and secretion of your cortisol, so they are in parallel. And again, I will reiterate that the highest point of ACTH secretion and cortisol synthesis and secretion is right after you wake up. After that, the levels steadily decrease to reach a nadir or lowest point at around 12 midnight. And while you sleep, the levels rise up again to reach the highest point or peak right before you wake up. Then, pababa ulit. And again, if you alter your sleep-wake cycle, you can alter this diurnal pattern as well with deleterious effects because you will have high cortisol during times when normally it is low. Okay, so we'll be discussing cortisol effects, and for effects, you therefore have to discuss its receptor. So the receptor of your glucocorticoid, which is the glucocorticoid receptor, when unbound, is found in the cytoplasm. Again, it is kept in the cytoplasm by a chaperone protein, which is your HSP, or your heat shock protein. Once your cortisol reaches the target cell, being a steroid, it traverses the cytoplasmic membrane and will bind to your glucocorticoid receptor. So you have one on this side and one on the other side. Binding of your cortisol to the glucocorticoid receptor causes dislodging of the heat shock protein. And therefore, this allows your hormone-bound glucocorticoid receptor first to form a dimer with another hormone-bound glucocorticoid receptor. And then this dimer will translocate to the nucleus. And via zinc fingers shown here, will bind to GREs, which are glucocorticoid response elements, which are key regulatory areas in your DNA, which will cause an increase or decrease expression of certain genes, which will cause an increase or decrease transcription of said genes, which will cause an increase or decrease synthesis of certain key regulatory proteins within that cell. And in so doing, therefore, again, it acts as a transcription factor once activated. And although we think of it in that manner, diba, the hormone-bound receptor translocates from the cytoplasm to the nucleus as a dimer and then will attach to GREs. But sometimes it's not that simple. Sometimes instead of the activated hormone-bound glucocorticoid receptor attaching to these GREs directly, sometimes certain books or articles will say, as in endotext.org, that actually it will attach and activate a transcription factor. And hence, this glucocorticoid receptor does not directly attach to the DNA DAO, but rather it attaches and activates this transcription factor, which in turn activates or represses transcription of certain genes. And of course, for this transcription to be initiated, you have these co-activators and you have to form the transcription initiation complex. So, there are many ways by which your hormone-bound glucocorticoid receptor can activate or inhibit transcription. So it can bind directly to your GREs. It can be tethered to the DNA via a transcription factor, or it can act synergistically with another transcription factor and promote transcription. So, we'll discuss effects of your cortisol, and again, your cortisol is a stress hormone. So think of all the things you need when you're stressed. Your glucocorticoids were so named because early on they recognized that cortisol increased blood glucose levels. Okay, so it is involved in glucose homeostasis, and therefore it is fitting, I think, that we discuss first the metabolic actions of your cortisol. Cortisol is critical for survival and during fasting. It promotes an increase in glucose and it is diabetogenic. It increases your gluconeogenesis and decreases insulin-mediated glucose uptake in your insulin-sensitive tissues, which are your muscle and adipose. At the same time, it promotes lipolysis and ketogenesis. It also increases protein catabolism for more amino acids for gluconeogenesis. And again, it is diabetogenic, lipolytic, and ketogenic. Cortisol has potent anti-inflammatory effects, which is why I'm sure you know of people or you yourself have taken synthetic glucocorticoids for inflammations or allergies. This is based on the fact that we know that cortisol can inhibit various steps in the inflammatory response denoted with those green bars, and can also inhibit various steps in the immune response. Aside from its metabolic effects and its anti-inflammatory effects, other effects of cortisol in the body include the following. In the bone, it stimulates your osteoclasts while inhibiting your osteoblasts, and therefore it decreases bone formation while increasing lysis of old bone or bone resorption. In the skin, it decreases collagen and connective tissue formation in the skin. I already mentioned the anti-inflammatory and anti-immune response. For the cardiovascular system, it maintains cardiac output and increases arteriolar tone. It also maintains sensitivity of these tissues to catecholamines, epi and norepi. It maintains muscle function, but also decreases muscle mass. Remember that it promotes protein degradation. It modulates emotional tone and wakefulness. In the kidneys, it increases GFR and free water clearance. For people who are pregnant, glucocorticoids facilitate the maturation of the fetus, such that if you're a nursing student who have rotated at students who have rotated at the IMU or intensive maternal unit or have practiced there, you will note that certain high-risk mothers who are at risk for preterm labor or who are undergoing preterm labor, some of them are being given high-dose glucocorticoids, synthetic glucocorticoids, in an effort to mature the lungs of the fetus if they give birth prematurely. So if we put together all the effects of your cortisol, you will get a table or picture like this from Netter that you will review at your leisure. Do you have questions regarding your cortisol? The silence is frightening. Okay. Sige, pag-isipan mo muna. Okay. I'll move on to your adrenal androgens, which are also products of your fasciculata and mid-ocularis layers. So again, we begin with pregnenolone. Pregnenolone is converted to your progesterone via your 3-beta-hydroxysteroid dehydrogenase. Both your pregnenolone and progesterone can be converted to your 17-alpha-hydroxypregnenolone and 17-alpha-hydroxyprogesterone, respectively, via the enzyme 17-alpha-hydroxylase. These two, in turn, can be converted to your dehydroepiandrosterone or DEA and your androstenedione, respectively, via your 1720 desmolase, which some books have recognized to be the same as your 17-alpha-hydroxylase. Your DEA can be further sulfated to form your DEAs, or dehydroepiandrosterone sulfate. Together, these three, your DEA, DEAs, and androstenediones, form your adrenal androgens. Of these three, it is the DEAs, which is the adrenal androgen, which is secreted in the greatest amount. These adrenal androgens are far less potent than gonadal androgens, but can still contribute to the circulating testosterone pool because in the periphery, you can convert these to your testosterone via your 17-ketosteroid reductase in the periphery. Your adrenal androgens, once they are secreted into the plasma, bind weakly to albumin. Diba, steroid hormones sila. But the binding to this transport carrier protein, albumin, is weak. This is in contrast to your potent androgen, which is your testosterone from your gonads, which is bound extensively to its own carrier protein, which is the SHBG, or sex hormone binding globulin. Adrenal androgens are metabolized in the liver into these products, androsterone and ethiocolanolate, which are excreted in the urine. These byproducts of androgen metabolism are not specific to the adrenals, meaning if you degrade the testosterone or metabolize testosterone produced by your gonads, you will also produce these byproducts in the urine. In contrast, your DEAs is also excreted in the urine. It is specific to the adrenals, and therefore, if you measure these urinary 17-ketosteroids, meaning your urinary DEAs, androsterone, and ethiocolanolate, it is the urinary DEAs which will tell you if that androgen actually originated from the adrenals or not. Again, your adrenal androgens are weak androgens, and if you want them to become more potent, you would need peripheral conversion to testosterone. Of themselves, however, males, would you miss them? No, because I presume you have two balls, which are the predominant source of your testosterone. And therefore, in males, adrenals only contribute about 5% of your circulating testosterone or androgen. And therefore, they are unimportant as compared to the testes as an important source of testosterone. So males would not miss it. Females, would we miss it? We would probably not miss it because, until we reach puberty, we keep waiting for all our hair to come out. Para masabi na nagdadalaga tayo. But once they arrive, we spend all of our time trying to get rid of axillary and pubic hair. I mean, am I right? Okay. So would we miss it if it's gone? Because its effect in females is to sustain normal axillary and pubic hair. Would we miss it? The realities probably will say no. But we will definitely know that it is gone. Okay? Kasi wala na tayong axillary or pubic hair. Okay. In females, it also maintains RBC production. Do you have questions regarding your adrenal androgens? Short but sweet lang. They say that your adrenal androgens are but mere byproducts of cortisol formation. Kasi pag tinignan natin at inisip natin, hindi ganon ka-importante, parang lumalabas. Except for females, for the pubic and the axillary hair. It's not that important. So some people actually say that, yeah, these are byproducts of cortisol formation. Okay. So, walang tanong? Okay. From the fasciculata and reticularis, I'll move on to your glomerulosa, which synthesize your mineralocorticoids. In the glomerulosa, you do not have substantially the 17-alpha hydroxylase enzyme, and because of that, you cannot form the adrenal androgens or the cortisol. All the pregnenolone is preferentially shunted towards the formation of your aldosterone. Also, in your glomerulosa, this is the only place in your adrenal cortex that you find the enzyme aldosterone synthase, which catalyzes the final step in the formation of your aldosterone. For all of these reasons, therefore, it is only the glomerulosa which is capable of synthesizing and secreting your aldosterone. Again, as with all steroid hormones, there's no storage pool for aldosterone. It will only be synthesized once you need it. So again, we start out with your pregnenolone, which is converted to your progesterone via your 3-beta hydroxysteroid dehydrogenase, converted to your 11-DOC or 11-deoxycorticosterone via your 21-alpha hydroxylase, in turn converted to your corticosterone via your 11-beta hydroxylase, and finally to your aldosterone via your aldosterone synthase. To discuss regulation of your aldosterone would not be complete if we do not review again what you need to review for renal physiology. So I'm sure Dr. Arabe already lectured this to you. Okay. So this is your JG apparatus. This consists of the area where your distal tubule comes in close approximation to its own glomerulus. And your JG apparatus consists of these specialized cells. First is your granular or juxtaglomerular or JG cells, which are specialized modified cells of your afferent arteriole. They're responsible for detecting pressure changes and volume delivery to your glomerulus and are also responsible for synthesizing and producing your renin. Specialized cells of your distal tubule are known as your macula densa. These are also part of your JG apparatus, which detect changes in sodium chloride delivery to the glomerulus. As I'm sure was discussed by Dr. Arabe, there are two things which are potent regulators of aldosterone secretion. The two major secretagogues, that which cause the secretion of your aldosterone. First is an increase in plasma potassium. And second is your angiotensin II, which is a potent vasoconstrictor, but also a major secretagogue, that which causes the secretion of aldosterone. Between these two, however, it is the angiotensin II which is really the major cause for secretion of your aldosterone. Your angiotensin II acts via a G-protein coupled receptor, which uses phospholipase C as an effector. This is your angiotensin I receptor. Okay. So with your plasma potassium, it's relatively easy enough. You have an increase in plasma load. This increases your plasma potassium. This causes depolarization here in your zona glomerulosa, which causes an increased synthesis and secretion of aldosterone. For your angiotensin II, just as a review, when you have anything that decreases your extracellular fluid volume, whether it be you lost a lot of fluid, you bled out in hemorrhage, you assumed an upright position, or you deprive yourself of sodium, this decreases the ECF volume. The decrease in ECF volume triggers or stimulates your JG cells, which then secrete and produce your renin. Your renin is both an enzyme and a hormone. Your renin stimulates the conversion of this protein from your liver, the angiotensinogen. Renin will catalyze its conversion to the active angiotensin I, which is rapidly converted to your angiotensin II via this ACE, or angiotensin converting enzyme, from your lungs. This angiotensin II is a potent vasoconstrictor, which restores your blood pressure and also stimulates your zona glomerulosa to synthesize and secrete your aldosterone. Other modulators of secretion, whether for stimulation or inhibition, are listed on the board. Please note that ACTH there is but a minor modulator. What does that mean? Even if you have a pituitary problem, if your adrenals are intact, if you have a lack of ACTH, you will not be able to produce sufficient cortisol or adrenal androgens, kasi ACTH is important for that. But if your adrenal gland is working properly and it is only the pituitary only, if it is only the ACTH which is the problem, you will still be capable of synthesizing and secreting aldosterone as long as you have the RAAS system in place. Because as long as your angiotensin II and your kidneys are working, you can still stimulate the synthesis and secretion of aldosterone. The same is true for hyperkalemia or high potassium. Secreted aldosterone, 37 to 50% of it remains free in the plasma. The rest are weakly bound to transport proteins, which are listed on the board. Your cortisol binding globulin, your aldosterone binding globulin, and your albumin. Aldosterone is degraded, 90% cleared in the liver, reduced to tetrahydroaldosterone, which is then conjugated to become glucuronides, which are then excreted into the urine. So before we discuss the effects of aldosterone, which I'm sure you already know from the renal physiology lecture, I'd like to delve some time on the mineralocorticoid receptor, because this is the only way that your aldosterone can cause effects in the target cell, which is mainly the principal cell in the kidneys. Your mineralocorticoid receptor is when unbound, is a cytoplasmic receptor. It is very similar to your cortisol's receptor, which is your glucocorticoid receptor. And in fact, this mineralocorticoid receptor is sometimes called your type 1 glucocorticoid receptor. So, when unbound, the aldosterone receptor or mineralocorticoid receptor is in the cytoplasm. Once aldosterone binds to it, the complex then translocates to the nucleus and will bind to SREs, or steroid response elements, which are key regulatory areas in your DNA to affect gene transcription. I would like you to note that although we try to simplify things and tell you that each receptor is specific for a specific hormone or ligand, sometimes that's not entirely true. This one, case in point, no? Your mineralocorticoid receptor has equal affinity to your aldosterone and cortisol. It has equal affinity to your aldosterone and cortisol. And if you think about the fact that the circulating levels of cortisol are much higher than the circulating levels of your aldosterone, you might begin to worry. Because iba naman ang effect talaga na desired natin with cortisol, right? But if that cortisol reaches the kidneys and binds to the mineralocorticoid receptor there, to which it has equal affinity to, then, even if it is cortisol, it will cause all the effects of aldosterone, which we might not want at that particular time. So how are we protected from these mineralocorticoid effects of cortisol? If you will recall, this enzyme which I told you about, this 11-beta-hydroxysteroid dehydrogenase, now, in the target cell of your mineralocorticoid aldosterone, specifically your principal cells in the kidneys, you have this enzyme. the 11 beta, type 2 11 beta hydroxysteroid dehydrogenase, which converts your cortisol to cortisone. Now, fortunately, here in your kidneys, once your cortisol is converted to your cortisone, like a lock and key, does not have affinity to that mineralocorticoid receptor. And therefore, by converting your cortisol to your cortisone, this actually confers the specificity of action of the mineralocorticoid receptor to aldosterone. Bakit? Kahit mas marami yung cortisol na nandun, nako-convert naman siya sa cortisone in this principal cell, which is the main target cell of your aldosterone. So kahit nandun yung aldosterone, mako-convert naman siya mainly to cortisone, and that cortisone will not bind to mineralocorticoid receptor. And therefore, the mineralocorticoid receptor is kept free for binding with aldosterone. This is important because later on, you will realize that if you have excess cortisol, like in Cushing syndrome that we will discuss later, this will overwhelm all the type 2 11 beta hydroxysteroid dehydrogenase in your kidneys. Kasi may hangganan lang naman yung number na yan, di ba? And if you overwhelm the number of this enzyme, not all of those excess cortisol molecules will be converted to cortisone. And therefore, some of those cortisol molecules which were not converted to cortisone will attach and activate a mineralocorticoid receptor. And in so doing, in the presence of excess cortisol, you can get these mineralocorticoid or aldosterone effects, even if it's just cortisol. But it's just physio. Kasi minsan tinatanong sa amin yun, panag-rounds kami. Patients, when I was a fellow, we would make rounds, and then the endocrine consultant would ask, oh, ayan, that patient has Cushing syndrome. How come you have some of the symptoms which are referable to aldosterone? So, syempre sabi mo, ma'am, because in excess, cortisol can have mineralocorticoid effect. And then comes the dreaded question, how? Hindi ko masagot. Kasi, I forgot to read that night. Then, you go home and, I'm so stupid. Diba, you realize na yun lang, simple ang sagot. It was just physiology. Even if we think of the mineralocorticoid receptor, once it is activated and bound to aldosterone, it will translocate to the nucleus and bind to SREs or steroid response elements there. Sometimes, your mineralocorticoid receptor does not bind directly to the DNA. Instead, it can bind to transcription factors instead, producing these genomic actions or effects via protein-protein synergy, not by directly binding to the DNA. And although, sana ganun lang kasimple na all the effects of aldosterone were via genomic actions, meaning via affecting gene transcription, diba? But it's not that simple. Because they have realized that there are certain effects of aldosterone which do not take hours, diba? If you affect gene transcription, this will take hours to days. But there are certain effects of aldosterone which only take minutes. And because of this, they have realized that for these effects to take only minutes for aldosterone, there are non-genomic effects of aldosterone, meaning that there are ways that aldosterone can cause effects in its target cell, not by involving this nuclear receptor, not by involving gene expression, but by non-genomic effects. It probably binds to a different receptor daw, or affects activity of certain channels. It can bind out to another cytosolic receptor that doesn't translocate to the nucleus or to a membrane-bound receptor. However, these effects of aldosterone, which are non-genomic, are transient, and we do not know the significance of these effects yet. The reason I tell this to you, even if we want to make simple for you, because when you're reading, mamaya may mababasa kayo na non-genomic effects of the steroid hormones. This is one way by which this happens. So, as a review for the aldosterone, renal effects are as follows. So, you have your aldosterone, which binds to your mineralocorticoid receptor in the cytoplasm. The hormone-bound receptor translocates to the nucleus and binds to key regulatory areas here to affect gene transcription, binds to your SREs, your steroid response elements, para simple na lang. And because of this, will increase or decrease the transcription of certain genes. In effect, it will actually increase the synthesis of these key proteins, which are your SGK, your serum glucocorticoid inducible or stimulated kinase, your Kirsten-Ras, and your CHIF, which is your corticosteroid hormone-induced factor. These three proteins, SGK, Kirsten-Ras, and CHIF, affect the activity, expression, and synthesis of various channels and affect the permeability of this principal cell, both in the apical and basolateral membrane. By affecting the activity, synthesis, and expression of certain of these ion channels, it is in this way that aldosterone affects sodium reabsorption in your kidneys. The main effect of your aldosterone in your kidneys is what? To reabsorb sodium in the principal cell through the principal cell in exchange for secretion of potassium. So how is this being accomplished by aldosterone? So let's orient ourselves. This is the principal cell, which is the target cell of your aldosterone found in the collecting tubule and DCT. So to orient ourselves, this is the tubular lumen where the urine is being formed. This is your apical membrane. This is your principal cell. Here, you have your basolateral membrane, and here you have the circulation or blood. So how do we promote sodium reabsorption in the presence of aldosterone in your principal cell at the DCT and collecting tubule? First, aldosterone increases expression, synthesis, and activity of this epithelial sodium channel found in the apical membrane. This facilitates the uptake of sodium from the tubular fluid into the principal cell. Next, the sodium is now inside your principal cell, right? You need to bring it to the blood. This is accomplished because aldosterone increases synthesis, activity, and expression of the sodium-potassium ATPase, which is found in the basolateral membrane, which brings out the sodium from the cytoplasm of the principal cell into the blood. In exchange for sodium, however, you get a potassium ion from the blood, which then enters the principal cell. This potassium will be extruded out in the presence of aldosterone to the tubular fluid to be peed out in the urine. How? Because in the presence of aldosterone, you increase the potassium permeability of your apical membrane. It becomes more permeable to potassium, such that the potassium that you got from the blood through the sodium-potassium ATPase is now extruded out via the apical membrane to the tubular fluid. Your aldosterone also increases synthesis and activity of your SGK, which is your serum glucocorticoid stimulated kinase, or SGK. Your SGK increases activity and synthesis and expression of your epithelial sodium channel in the apical membrane, which I discussed earlier, and also further enhances the permeability of your apical membrane to potassium, thereby again facilitating sodium reabsorption and potassium excretion. Lastly, your aldosterone also enhances activity and synthesis of your CAP, which is your prostatin, also known as your channel activating protease, which activates this epithelial sodium channel in the apical membrane, which again facilitates sodium reabsorption from the tubular fluid into the principal cell. Net effect of all of these on your apical and basolateral membrane is to promote sodium reabsorption in exchange for potassium. So you reabsorb the sodium from the urinary tubular, from the urine, you reabsorb the sodium, and with it, water will always follow. In exchange for this reabsorption of sodium, you secrete out the potassium. Such that in the presence of aldosterone, if you check the urine has high potassium levels but low sodium levels. And if you check the plasma, your plasma potassium will go back down to normal, and you're retaining more sodium in the plasma. Since you retain the sodium, water will follow because of osmosis. So in the presence of this retention of sodium, water will follow. You thereby increase your extracellular fluid volume, thereby restoring your blood pressure back up to normal. If you will recall what prompted the secretion of aldosterone in the first place, it's two major things, diba? An increase in plasma potassium and a decrease in extracellular fluid volume. In the presence of aldosterone, once you've reduced your plasma potassium back down to normal, and once you've increased the ECF volume back up to normal, you remove the stimulus for the synthesis of aldosterone. And as such, once you get what you want, which is homeostasis, meaning a return of your plasma potassium and extracellular fluid volume back to normal, you will reduce secretion and synthesis of aldosterone. Again, always recall na no hormone is ever produced at zero rate. There's always a basal rate. It's just a matter, do we produce more or do we produce less? So in the principal cell, we said that there is a reabsorption of your sodium and a secretion of your potassium. Since you keep reabsorbing all the sodium in your principal cell, the tubular lumen becomes even more negative because you're losing even more positive charges, diba? From the tubular lumen, you're losing it to the blood. So if you're losing a positive ion from the tubular lumen, your lumen becomes even more negative. So diba, opposites attract. Since the lumen is negative, it will draw out positive charges. In the principal cell, we already said it will draw out the cation potassium. However, in the intercalated cell, what is drawn out is your H+. Okay? You have the same effects in the presence of aldosterone on your sweat glands and salivary glands. Meaning, in the presence of aldosterone, when you check your sweat and saliva, it will contain less sodium, more potassium, and probably more H+. In the colon, it also enhances sodium absorption. The net effect of all of these is what? To conserve sodium and with it water.